Welcome back to The Explainer. Today, we're zooming in on one of the most incredible biological races against time in existence, the human zygote's epic 7-day journey from fertilization all the way to implantation. I mean, we literally all start as a single cell, right? But the sheer amount of highly coordinated, mind-blowing biological engineering that has to go down in just one single week, it's absolutely wild. So if you're a focused learner looking to totally master the key stages of early human development, you are in exactly the right place. Okay, let's dive into this highly structured study guide. Here is our agenda for today. First, the starting line. Then we'll go from morula to blastocyst. Third, we'll hit the VIP cell layers. Fourth, forming the inner cell mass. And finally, implantation and its complications.
Section 1. The starting line. The first hours of cellular division.
So our hero's journey kicks off right after fertilization, deep inside the fallopian tube. To give you a clear chronological roadmap of the human embryo's very first week, let's take a look at this timeline. Right at hour 0, we have fertilization. By 30 hours, boom, that single cell has divided into two. Fast forward to day 3, and we hit the morula stage. By day 5, it transforms into a blastocyst, and finally, right on day 7, it reaches its final destination for implantation. So let's break down how this whole thing builds, starting with those very first cell divisions.
The very first major biological process at play here is called cleavage. Now, you probably know cleavage is a specific type of rapid mitotic cell division. The cells are multiplying super quickly, but here's the catch. They actually skip the G1 and G2 growth phases that you'd normally see during standard cell division.
And this brings up a brilliant comparison between these two biological processes. In normal mitosis, the cell size actually grows between divisions, but in cleavage, the number of cells increases rapidly. We're talking 2, 4, 8, 16, but the overall size of this entire structure remains absolutely identical to the original single-celled zygote. All these brand new cells, which we call blastomeres, they're still fitting snugly inside that exact same original protective shell.
Because the overall size isn't increasing, we end up with this totally fascinating biological paradox. With every single division, the nuclear material, the DNA, is doubling. But the amount of cytoplasm inside that protective shell stays perfectly constant. As a result, the nucleocytoplasmic ratio shifts dramatically upward.
By the time we hit the 16-cell stage, the ratio of nuclear material to cytoplasm has spiked significantly compared to our single-celled starting line. The cells are getting smaller and smaller, literally packing more and more genetic material into the exact same volume of space. It's incredibly efficient.
Moving right along to Section 2, morula to blastocyst, covering day 3 to day 5 of the journey.
As this really dense cluster of dividing cells journeys down the fallopian tube, it hits its next big milestone around day 3, the morula stage. Fun fact, the term morula actually comes from the Latin word for mulberry, and if you look at it, the shape perfectly resembles one. It's a solid, tightly packed ball of 8 to 16 cells. At this point, there's zero empty space inside. It is just a completely solid, dense sphere of blastomers.
But as soon as it hits the 32-cell stage, this amazing transformative process called cavitation kicks in. Fluid secreted by the endometrium, often called uterine milk, starts to actually seep inside this solid cell mass. This fluid physically pushes the cells apart to form a hollow cavity right in the center, which is known as the blastocell. So the once solid morula has officially hollowed out to become a blastocyst.
All right, Section 3, the VIP cell layers, the zona pellucida and the trophoblast.
During this hollowing out phase, the cells start organizing themselves into highly specific layers, essentially acting as two crucial character archetypes for our story. First up, we have the zona pellucida. This is that original protective shield we mentioned earlier. It's a thick, non-cellular glycoprotein layer that has literally been wrapping around the dividing cells since fertilization. Its main gig? Keeping the dividing cells safely contained and, absolutely critically, preventing them from randomly sticking to the walls of the fallopian tube while they're traveling.
The zona pellucida is supposed to stay totally intact until the embryo safely reaches the uterus. But what happens if this protective shield breaks prematurely while it's still in transit?
Well, here is the crucial point. If the zona pellucida ruptures too early, the embryo will just implant wherever it happens to be at that exact moment. Most commonly, this means it implants directly into the narrow walls of the fallopian tube, which results in an ectopic pregnancy. Because the fallopian tube simply cannot stretch and support a growing embryo the way a uterus can, this is a highly dangerous, life-threatening complication.
Our second VIP layer is the trophoblast. During cavitation, the outer ring of cells flattens out to form this specific layer. Think of the trophoblast as our master builder and communicator. It has a seriously busy functional role. It forms the outer boundary of the blastocyst. It eventually secretes specific lytic enzymes to literally hatch out of that zona pellucida. It forms the structures that will later become the placenta, and it secretes a vital hormone just to keep the pregnancy viable. We're going to return to that hormone in just a second.
Section 4. Forming the inner cell mass, the cells that become the embryo.
So while the trophoblast is busy managing all those exterior logistics, a really crucial cluster of cells pools together at one distinct corner, or pole, on the inside. This is the inner cell mass, and this tiny cluster is destined to eventually become the entire human body.
Now, what's really interesting about this slide is the miraculous potential of these early stem cells. The inner cell mass is absolutely packed with totipotent stem cells. Basically, at this early stage, these cells possess the miraculous ability to divide, differentiate, and give rise to absolutely any tissue or organ in the human body. They hold the complete, unedited blueprint for human life. It's just wild to think about.
Finally, Section 5. Implantation and complications. Reaching the final destination on day 7.
By day 7, our blastocyst finally reaches the safety of the uterine cavity. But the journey isn't quite over. It has to secure a permanent lifeline. Around day 5, the blastocyst officially hatches. The trophoblast secretes those lytic enzymes we talked about, which eat a hole right through the zona pellucida, and the pressure from the fluid inside literally forces the blastocyst to squeeze on out. Now completely free of its shield, by day 7, the blastocyst touches down on the uterine wall, the endometrium. And those outer trophoblast cells begin to actively burrow and invade the maternal tissue, essentially locking the two together in the process we know as implantation.
To pull off this invasion effectively, the hard-working trophoblast literally divides its labor, splitting into two distinct layers. The inner layer, called the cytotrophoblast, keeps its distinct individual cells. But the outer layer, the syncytotrophoblast, it actually loses its cell membranes, fusing into one giant multinucleated layer. This outer layer grows these powerful finger-like projections called chorionic villi, which physically eat into the endometrium to establish what will eventually become the placenta.
And once it's securely implanted, the trophoblast immediately starts secreting a chemical signal, human chorionic gonadotropin, or HCG. This hormone travels straight through the bloodstream to tell the ovaries, hey, we made it, keep producing progesterone, do not start a new menstrual cycle. Fascinatingly, because HCG is produced exclusively by the early embryo's trophoblast cells, it is the exact hormone that standard over-the-counter pregnancy tests are looking for to confirm a pregnancy.
It is a totally flawless, deeply intricate process, all wrapped up into just 7 days. But it really leaves us with this incredible thought. We all start as a single microscopic cell. But how does that one cell possess the perfect biological clock to know exactly when to divide, exactly when to form a cavity, and exactly when to hatch? It is a true miracle of human development that scientists are still studying to this day. Thank you so much for joining me for this explainer. Keep asking those big questions and keep learning.